Please join me in welcoming Dr. Allison Alberts.
Thank you, Janet. You know, I was looking at this fabulous brochure about the upcoming talks. I don't know how many of you have had a chance to look, but did you see that there's one coming up on chocolate? And they're actually going to serve chocolate. And I thought, well, what could I serve that would go with my talk? And dead whales, just not going to cut it. So anyway, thank you so much for coming. I'm excited to tell you about the work that goes on at the Arnold and Mabel Beckman Center for Conservation Research, which is located at the San Diego Zoo's Wild Animal Park. Our Beckman Center houses CRESS, which stands for Conservation and Research for Endangered Species. Our mission is to generate and apply scientific knowledge to help conserve species, both here in California and also around the world. I'd like to spend about the first 10 minutes or so just telling you a little bit more about the work that goes on at our Beckman Center, but the main part of my talk is going to be to share with you the incredible story of one species, the California condor. I want to begin by sharing just a little bit about the history of CRESS. CRESS was founded in 1975 by Dr. Kurt Benerschke, a medical doctor from Germany who had a true passion for wildlife. The first CRESS lab was actually located in Balboa Park in downtown San Diego in pretty Spartan conditions. The quarters that we were housed in were left over from the World's Fair, which took place in 1915 and 1916, so the buildings were very old. And the original mission of CRESS was to improve the genetic and reproductive health of the zoo's extraordinary animal collection. In the early years, the zoo had to get pretty creative about how to fund CRESS's efforts. Luckily for us, some of you may remember that Dr. Benerschke had a son named Rolf, who was a really talented place kicker for the Chargers in the 1970s. So in collaboration with his dad, Rolf started a program called Kicks for Critters, in which he would recruit football fan donors that would make a contribution to CRESS every time he kicked a field goal. And the program actually ended up raising several $1 million to support endangered species research. So I have to say, go Chargers, and in light of what's coming up this weekend, good luck, Chargers. In the 30 years since CRESS was founded, we've grown significantly, first by adding a much greater variety of scientific expertise, and more recently by expanding our efforts to include field conservation programs in more than 35 countries. Today we've got a staff of over 120 people, making us the largest research team housed at any zoo in the world. In 2004, we moved from the zoo to the Arnold and Mabel Beckman Center for Conservation Research, which was funded by a generous matching grant from the Beckman Foundation. Our Beckman Center is 50,000 square feet in size, and it's located adjacent to Harder Hospital, which is one of the largest and most advanced veterinary facilities of its kind in the world, and together they form the San Diego Zoo's conservation campus. The Beckman Center contains 6 large state-of-the-art laboratories, 48 offices, a library, and a conference room. One of the things we're really proud of is the fact that the Beckman Center is one of the few laboratory facilities of its kind to be awarded silver-level LEED certification from the U.S. Green Building Council. And many of the features of the center led to this design. But 2 of the most important are the fact that 15% of all of our electrical needs are met by solar power. And in one of my personal favorite, all of our wastewater is treated on site, and then it's used to irrigate the large animal enclosures at the Wild Animal Park. At the Beckman Center, biodiversity conservation is really the theme that unites our work, and scientific innovation is the way that we achieve our success. And we like to say that we're the science of saving species. Our research team consists of scientists and technicians in 6 research divisions. We carry out research in the areas of ecology, behavior, genetics, reproductive physiology, wildlife diseases, and we've recently established a new division that's devoted to plant conservation. Our Applied Animal Ecology Division does a lot of work doing field projects around the world. their emphasis is on reintroducing species that have been bred and reared in captivity back out into the wild. And they've worked to understand the social and ecological factors that are going to make a given reintroduction successful, and then to design the best methods for monitoring the animals after they're released back out into the wild. And this slide just shows one of our researchers releasing a critically endangered Jamaican iguana back into its natural habitat. And I had to put some iguanas in my presentation because, as Janet said, I'm a big iguana fan. Our behavioral biologists try to figure out what environmental conditions are needed to successfully breed and manage animals in captivity. They've recently established a new sensory ecology program to better understand how animals communicate with each other. And they've got a strong focus on sound communication. One of the things they're studying is how mother okapis communicate with their babies. And we've discovered that they actually use infrasounds, which are so low in frequency that humans can't hear them, but the okapis can.
Excuse me, what species was this?
The okapi. Our genetics division provides information that helps guide captive breeding programs and also the genetic management of wild populations. A major focus for them is studying the genetic factors that are associated with susceptibility and resistance to disease in endangered species. And a big part of their work is growing our one-of-a-kind frozen zoo. The frozen zoo currently contains more than 8,000 living cell lines, eggs, sperm, and embryos from almost 700 species, including one extinct species, the pu'u'uli bird from Hawaii. It's the largest genetic resource of its kind in the world, and it's an important source of samples that we send out to other researchers who do studies not only on animals, but also work that benefits humans as well. Our reproductive physiology division uses innovative technologies that have been developed for humans and domestic animals and applies them to reproduction of endangered species. For example, they used a combination of ultrasound and infrared thermal imaging to visualize the tiny fetus when our giant panda Bayun got pregnant for the first time. And it's a big challenge to visualize a panda fetus because it's literally about this big when it's born. They're very, very small. But what they were able to do with this work was to accurately predict the exact day that she would give birth to her cub. Our wildlife disease labs consist of a pathology lab, a clinical lab, a molecular diagnostics lab, and a histology lab that all work together to try to stop the spread of diseases and to make sure that our reintroduction candidates are healthy enough to be released back out into the wild. They're also really interested in how diseases are transmitted between people, domestic animals, and wildlife. And two of their current projects involve developing a vaccine program for West Nile virus and also creating a plan to prepare for the potential arrival of avian flu in North America. Just last year, we added a new division at CREST that's devoted specifically to plant conservation. Projects in this division include conservation and restoration of ecosystems, banking of seeds from rare species, and habitat monitoring and management. The division is currently collaborating with Kew Botanical Gardens in England to bank seeds from 450 of the most biologically and culturally important plants in San Diego County. Finally, the Beckman Center houses a conservation education lab. And the goal of this lab is to give students of all ages and their teachers the opportunity to interact with CREST researchers and provide them with experiences that will connect them with the science behind conservation. Over the summer, we hosted this enthusiastic group of 7th graders who came in and solved the DNA mystery, we call it furry forensics, and they used the same equipment and research tools that we use in all the labs at CREST. So far, the youngest visitors to the Conservation Education Lab have been this group of 2nd graders. And what they did was learn all about the different ways that we use radio telemetry to track animal movements in the wild. And you can see the blue, this is actually one of the antennas that you can use to pick up the radio signals. So kids had a lot of fun with that. So now I'm going to share some pretty sobering statistics that put the work we do at the Beckman Center into perspective. Since the time that I was born, the number of people on Earth has doubled. And by the year 2035, it's projected to double again. Half of the planet's land has been impacted by man. More than 16,000 species are threatened with extinction, including almost a quarter of all mammals, 12% of all birds, half of all reptiles, a third of all amphibians, and perhaps the most concerning of all, 70% of all plants. Now, before you get too depressed, I want to emphasize that there is some good news to be had. There's a number of conservation organizations around the world, including CREST, that are making a real difference. What makes CREST different from other conservation organizations is that we're committed to using the best possible science to rescue the most critically endangered species from the brink of extinction. And in many cases, these are species that other organizations have given up hope of saving. And tonight I'm going to share with you the remarkable story of just one of those species, the California condor. By the mid-1980s, there were only a few dozen condors left in the world, and we know that their decline was a direct result of lead poisoning. Because condors are scavengers, they often feed on carcasses that are left behind by hunters, which may still contain the lead ammunition used to hunt them. When condors ingest fragments of lead bullets and lead shot, they suffer serious neurological problems, just like people do, including death. And by 1987, the problem had become so severe that only 27 birds were known to still be alive. And the U.S. Fish and Wildlife Service made the controversial decision to bring them all into captivity. And now, at that time, there were some vocal people who felt that saving the condor was a lost cause and that the species should be allowed to die with dignity in the wild rather than be brought into captivity. There was also some resistance from the Chumash Indians, who believed that condors carry people on their wings to the spirit world after they die. So understandably, they were opposed to taking the last of the condors out of the wild. But the Chumash also understood that extinction was not an option. So one day, two biologists from the San Diego Zoo were thinking about this, and they came up with the idea of engaging the Chumash by asking them to name all of the condors. And the Chumash elders liked this idea, and they gave us a list of names like Gewi, which means to cast a shadow, and Topa Topa, which means rolling pastures of grain. So what our keepers would do is go over the list of names the Chumash gave us, then they choose the ones they could pronounce. And the Chumash have since become real supporters of the condor program, and now they actually have a special blessing ceremony that they do for every bird before it's released into the wild. So once everyone agreed that the best hope for the condor was to establish a captive breeding program, all 27 remaining California condors were captured, and they were brought into captivity at the Los Angeles Zoo and the San Diego Wild Animal Park. We became part of the U.S. Fish and Wildlife Service recovery plan to save the species from extinction. The plan called for us to establish 3 distinct populations of at least 150 birds each. Condors were to be reestablished first in California, then in the Grand Canyon in Arizona, and most recently in Baja California, Mexico. So fortunately, the condors did adapt really well to captivity, including pairing up and mating without really any problems. And here you see female condor Kachuma. She's unabashedly weighing herself in her enclosure at the Wild Animal Park. And the reason I include this picture is that you can really get an appreciation for the size of these birds. They're the largest bird in North America, and they have an incredible 9-foot wingspan. But even before the birds arrived, we'd already been involved in genetic screening of the remaining wild population. Scientists at the Beckman Center were actually able to work out a new technique for doing genetic testing of the birds without disturbing them. And what they would do is they could extract DNA from their molted feathers. And this work showed that there were 3 separate genetic clans, and based on it, we were able to best decide how to pair the birds to make sure that genetic diversity would be lost over time. Later, we were able to develop a technique to sex the chicks using just a tiny droplet of blood that could be collected from the eggshell after the chicks had hatched. So luckily for us, condors exhibit a behavior called double clutching. And what this means is if you take away their first egg, they'll actually lay a second egg within 25 to 30 days. So when an egg is laid, the keepers remove it and they put it through a procedure called candling. And then the bright light of the candler allows them to tell whether the egg is fertile or not. So we then remove the eggs and place them in an incubator for the next 57 days. We candle each egg every day to make sure it's developing properly. Because wild condors usually only fledge one chick every 2 years, the practice of removing the first egg has really allowed us to greatly increase the reproductive rate of the captive population. We then allow the adult pair to hatch their second egg naturally. So the parents, assisted by the keepers if necessary, will gently use their beaks to help the chick hatch. They then rear the chick together, alternating feeding duties between mom and dad, just like they would do in the wild. But condors can't raise more than one chick at a time. So what happens to that first egg that we remove for artificial incubation? Our keeper staff were able to design a very realistic-looking puppet that could be used to successfully rear the chicks and prevent them from getting imprinted on humans. So this is a real condor chick, and this is a puppet parent. When the chicks reach about a year of age, that's when we release them out into the wild. And I have to admit, they're not very graceful flyers at first. And landing again can be a bit of a challenge. But they do eventually get the hang of it. Well, not everything went as smoothly as we thought it would in the early years of the releases. It turns out that collisions with power lines were a real problem. And you can imagine what it would be like trying to navigate through something like this on a foggy morning, especially with a 9-foot wingspan. But fortunately, we were able to largely solve the problem by using aversion training before the birds were released. And what we would do is put up fake power poles in the condor zoo enclosures that would give them a slight shock whenever they tried to land on them. And it really made the birds not only avoid the power poles, but the wires as well. They just really didn't want anything to do with anything that was near a power pole. So the aversion training has reduced the collisions with power poles almost to zero. It's been a really successful management technique. The first release condors began to breed in the wild in 2003, but only about half of the chicks hatched in the wild have survived so far. At least 10 of them have died because of complications resulting from ingesting small pieces of trash. And what's happening is that the parents seem to be mistaking small pieces of trash for bone chips, which they would normally feed the chicks in the wild to help them ensure that they have enough calcium to grow. So as a result, the parents are inadvertently killing their own chicks. Now here's some examples of the types of trash materials that we found inside of chicks. Bottle caps, nuts and washers, shards of glass, and plastic. In one chick alone, our pathology staff found 19 bottle caps, 23 pieces of broken glass, 2 soda pop tops, and a piece of rubber. Fortunately for this chick in California, the biologist did arrive in time. You can see that they were actually able to go into the nest, reach down his throat, and remove a shard of broken glass. And in the end, the biologist removed 3 pieces of glass from the chick's crop, and you can see that at least 2 of them came from a discarded Pepsi bottle. Well, in studying the problem, we've learned that it's actually the female that tends to be the culprit. And we're working to address the problem by placing bone chips directly into the nests of wild condors that are rearing young. If we make sure that the breeders have access to the bone chips, it's really helped reduce the problem. But it hasn't completely eliminated it. We're also looking at aversion training to keep the parents from picking up trash in the first place, and also working to educate people who live near condor release areas about the importance of not littering. Lead also continues to be a problem for the condors. Often when birds die in the wild, we're never able to determine why, but we do know that lead poisoning has been responsible for about a quarter of all deaths of condors in the wild. And for this reason, we actually capture all of the wild birds at least once a year, and we take a blood sample, we test their blood for lead, and if lead levels are high, then the birds are brought into captivity and they're given a chelation treatment, which removes lead from the bloodstream. It works very well as long as you catch it in time, and then we're able to release them back out into the wild. So in 2000, the San Diego Zoo proposed to the Fish and Wildlife Service that we reintroduce California condors into Baja California in Mexico. And we know from these ancient Indian rock art paintings that condors existed in Mexico a very long time ago. And historical records show that they were still there until about the 1930s. And because human population density is so much lower in Baja than in the United States, we did expect that the lead and trash problems wouldn't be nearly as severe down in Baja. The site that we chose for the condors is located within the Sierra San Pedro Martir National Park. The site's in a really rugged, remote area that's within a few miles of the highest peak in Baja, El Diablo, which is at 10,200 feet. We do rely very heavily on the help of volunteers to keep the Baja Condor program going. We've got some great volunteer pilots that help us transport the condors. We put them in large dog kennels, and that works great. And we fly them from San Diego down to the Melling Ranch, which is just a few hours south of Ensenada. And the first birds arrived there in August of 2002. From the Melling Ranch, the birds were driven up the mountain to our on-site holding pen on the ridgetop. Their enclosure is about 60 feet by 60 feet by 30 feet high, and it's surrounded by nylon netting. What we were able to do is use living Jeffrey pine trees to hold up the netting, which provides a really natural environment for the birds. And the birds then remain in this pen for up to 3 months before being released to the wild. And what that does is it allows them to get used to the sights and the sounds and the climate of the release area. The enclosure contains a blind with several one-way glass windows that allow our biologists to observe the birds without the birds realizing that they're there. And just like at the Wild Animal Park, we have a scale in the enclosure that the birds actually like to perch on, so it allows us to get these regular weight measurements on them without ever having to catch them. All of our veterinary health checks are done at night, including vaccination for the West Nile virus, and that allows us just to disturb the birds as little as possible. They're much calmer at night. It's their roosting, quiet time. So our crew of Mexican field biologists and students live and work on the site, and that's so that they can always be available to care for the birds and also follow them after they're released into the wild on a continuous basis. And you can see that our field camp's not fancy, but if you love the mountains, it's a really pretty place to be. Now, things can get a little challenging in the winter when we usually get several feet of snow on the ground. I think last year they got 11 feet, actually. And being a high elevation species, the condors are used to the cold. They don't mind it at all. It's really the field biologists that tend to have a hard time with it. But they like the snowmobile. That's fun to ride around on. The other challenge we faced at the site is fire. In 2003, we had a fire come through the area, burned about 3,500 acres. And although it gave us a scare, the condors really weren't harmed or disturbed by it at all. We actually saw them. They were purposely making use of the thermal air currents produced by the fire, and they loved it. They would soar at greater heights than they would normally be able to do. So, fine for condors. So condors are highly social animals, and we house up to 4 young birds in the release pen at a time. And in order to instill proper condor social skills, the juveniles are housed with a mentor bird. So this is Gawi. She's our 15-year-old adult female mentor, and it's really her job to keep the youngsters in line and make sure that they behave themselves. And the way condors are, I'm sorry to say, without a mentor, it's pretty much like Lord of the Flies. So it's a good thing we have Gawi. So here Gawi is interacting with 2-year-old number 284, and she's actually using her foot to make sure that the younger bird knows its place in the social hierarchy. Because the terrain is so steep and condors... Oh, wait, didn't mean to do that. Let's go back. Can we go back? Yes. Because the terrain's so steep and condors can fly for many miles, it's really important for us to be able to keep track of them. And we do that with specialized technology. So before being released, we outfit every bird with a radio transmitter that gets attached to the wing. Each condor's radio transmitter has its own unique frequency. And using a receiver, our field biologists Juan Vargas and Catalina Porras can dial in each bird's frequency and then use a handheld antenna to find the general direction in which the birds can be found. For greater distances over rougher topography, we actually use a light plane with an externally mounted antenna for the same purpose. Most of the birds are also outfitted with GPS transmitters that can communicate with orbiting satellites in order to track the birds' locations remotely up to 15 times a day, which is really critical for us trying to work in this mountain habitat where you just can't drive around and follow them. So this schematic just shows you in a very basic way how the GPS system works. Data from the GPS tags on the birds go up to an Argos satellite system, which sends information on the birds' positions to one of 3 stations, depending on where they are around the world, Australia, Paris, or New York. And conveniently for us, the information then gets emailed directly to our desktops. And the great thing about the GPS transmitters is that they also give us information about the birds' flying altitude and also their flying speed, so we can get a good idea of exactly where they are in a 3-dimensional sense. So following release, the normal pattern for the birds is to take longer and longer forays away from the release site until they are flying up to several hundred miles in a single day. And in June of 2005, the GPS satellite data showed us that 3 of our released condors had actually flown to within 15 miles of the Mexico-U.S. border, which was pretty exciting. But the real surprise came last July when 3-year-old female 321 followed a group of turkey vultures on a 165-mile trip across the border. She showed up in Anza-Borrego Desert. These are her triangles. This is her way up here. She got that far. She actually stayed there for a day, and then our feeling is that she looked around, realized she was alone, because they like to be with other condors, and she actually, the GPS data showed us that she made a beeline straight back to the release site, all in one day, to get back with the other condors. But it was really exciting because it was the first time that a California condor had been seen in San Diego County in over 100 years. And we do think they will come back. Our released condors have now ranged not only north and south, but also east and west to both coasts in Mexico, including landing on this beach in San Felipe. And hence my joke about dead whales. Although we provide food for the released birds, we've been happy to see that they have also begun to forage naturally on their own. And historically, marine mammal carcasses were probably one of the most important food resources for condors of all. And released condors were actually observed feeding on this whale carcass for over 8 months. So that's what they do. But the Baja condors do seem to especially like hanging out in desert canyons like this one that have standing water and more mild temperatures. So the top bird in the social hierarchy of the 19 condors that have been released in Baja so far is male 261. And in 2004, we noticed he'd been spending an awful lot of time with the top female, female 218, including displaying to her for over a year, and it seemed likely that she might become his mate in 2005. One of the threats for free-flying condors is golden eagles, which are known to have harassed, injured, and even killed condors in the United States. And at about this time, a golden eagle had been seen attacking condors in and around the Baja release site, including female 218. So we grew worried about her when she returned a day after the other birds from a trip south, and it appeared that she had an injured foot. So we brought her to a clinic in Ensenada and took x-rays, and the x-ray fortunately showed no major problems, and she made a full recovery within 2 months. But the incident set her back socially. Remember, I said condors are very, very social birds, and it moved her from the top female in the dominance hierarchy to the very bottom. And as a result, male 261 didn't want anything to do with her anymore. So the immediately subordinate female to 218 was 220. She then becomes the new top female, and she's now most likely to breed with male 261. So he began spending a lot of time with her, giving her a lot of attention, and we noticed the two of them teaming up together against some of the more subordinate birds, which is a sign that they've become an established pair. Well, the soap opera continues. Just before the summer of 2004, female 220 lost all but one of her tail feathers, and we believe it was most likely the result of an attack by a mountain lion that we knew had been hanging around the release site because we'd managed to capture a picture of him with one of our remote cameras. So it took this female 4 months to regrow her tail, and she also lost her top position in the dominance hierarchy, and she also was rejected by male 261. So it wasn't immediately clear whether 7-year-old female 217, she's now the third, the next down in the hierarchy, was going to pair up with male 261. But as the 2007 breeding season started, the GPS satellite data began to show us that they were synchronizing their movements and behaving as a pair. In February, we noticed that they were each taking turns going to a cave. We knew it was somewhere on the east side of the mountains. And egg-laying season's usually between mid-January and the 1st of May. So we began to get pretty excited about the fact that this pair might actually produce a chick. It turned out that when the birds were inside the cave, the GPS transmitters weren't able to communicate with any satellites, so we actually had to use visual observation to detect the exact location of the cave. So look at the slide and see if you can pick out where the cave is. Did you get it right? Yeah? So they didn't really pick a cave that was easy to get to, unfortunately for us. Once we located it, we had to find a safe route up the mountain, which actually required some technical climbing ability and specialized equipment, and it took about 2.5 hours to make the trip up the side of the mountain. But we got there, and we knew we'd actually found the nest cave when we saw female 217 landing in it. So on March 24th, we were able to enter the cave, and we saw her egg for the first time. We were really surprised to see that what this pair had done was chosen to nest in an abandoned eagle's nest made of sticks, because condors almost always nest in the sand. But a few weeks later, we became very concerned when the egg didn't hatch by the time we expected it to. So because we feared the chick inside might be dead, we decided to bring this pair a healthy egg from a pair of condors at the Wild Animal Park, switch it out, and to give this pair, as first-time parents, some experience at raising a chick. So our biologist, Dr. Mike Wallace, climbs up the mountain with the precious egg in his backpack, enters the nest with the replacement egg, and female 217 literally attacked him. And he realized she had to be protecting something that was really important to her. And when he was able to see around behind Mom, there was a chick. 2 days old, we think. So we followed the chick's development over the next several weeks, and everything did seem to be going okay. But then the parents suddenly changed their foraging behavior, and we knew something that was really wrong. And when we checked the nest the next time, the chick was gone. And the only clue that we had as to what might have happened was that there was a large number of parasitic flies in the nest. They actually turned out to be a type of bed bug. And we know that stick nests do tend to harbor parasites, which is probably the reason condors normally nest in sand, and eagles frequently change their nest sites. They don't nest in the same place year after year. So whether this chick got overwhelmed by the flies or was possibly predated by a returning eagle, those are the two theories that we have about what might have happened. We'll never know that. But what we do know is that this chick died of natural causes. It didn't die of lead poisoning, and it didn't die by ingesting trash. So despite the unhappy ending to the Baja chick story, reproduction in the wild was still a major milestone for the Baja project, and it's one that we hope will be repeated again this year. 5 of the chicks that were hatched in California in the wild last year did fledge successfully, evidence that some of these new management practices, particularly with regard to the microtash problem, are working. Unfortunately, we did lose 1 chick in California as a result of the wildfires that swept across Southern California in October. That was one of the ones at the Hopper Mountain site. But here's a photo of a wild chick that successfully fledged in Arizona last year. And you know it's a wild chick because, look, Mom, no wing tags. Okay. And more good news. Just a few months ago, Governor Schwarzenegger actually signed a bill banning the use of lead ammunition throughout the California condors range in California. And that's huge. That will really help us with the lead problem. And it was actually his idea to put a California condor on our state quarter. And he's been a big supporter of the condor program ever since taking office. So we have to be thankful to him for that. With nearly 300 condors living today, up from 27 just 2 decades ago, I think we can say that the California condor is definitely one species that has been brought back from the brink of extinction, and we're really proud of our role in contributing to that success. So what I'd like to do in the last few minutes of my talk is talk about a few other projects. Over time, we've been able to expand our efforts to include other species besides condors. In 1985, no more than 5 pairs of the loggerhead shrike remained on San Clemente Island, and everybody thought extinction was probably inevitable. But our scientists became involved in an intensive captive breeding and release program that involves raising birds on San Clemente Island and releasing them, and today there are more than 100 San Clemente Island shrikes in the wild. As part of our Hawaii Endangered Bird Conservation Program, we've bred and released more than 650 individuals of 7 species back to the wild, and for the first time ever, released puaiohi, raised from artificially incubated eggs, are now reproducing in the Alakai Swamp on Kauai. The first giant panda cub to survive in the United States was Hua Mei, born at the San Diego Zoo, and there have now been a total of 4 baby pandas born at the zoo: Hua Mei, Mei Sheng, Su Lin, and Zhen Zhen, who just got her name last month when she turned 100 days old. That's the Chinese custom, and her name means precious, which she certainly is. But most exciting of all is that the first giant panda cub to be born at the zoo, Hua Mei, now has 4 cubs of her own in China. And as a result of our research, the number of giant panda cubs born at the Wolong Breeding Center in China has increased from 25 to more than 100. And last year, you can see this crop of 16 panda cubs that were born all at Panda Nursery School. So for half of all species of Caribbean rock iguanas, fewer than 200 animals remained in the wild in the 1980s. But by 2006, scientists from the Beckman Center had helped return more than 600 individuals of 4 species back into the wild through captive breeding and release programs. And thanks to our efforts, several islands on which iguanas had gone extinct now have surviving and thriving populations. Historically, cheetahs have been very difficult to breed in captivity, but intensive behavioral and hormonal research at the Beckman Center has led to the birth of more than 130 cubs at the Wild Animal Park, and we're now able to share our methods with other cheetah breeding programs around the world. But there are a lot more species that need our help. We've recently begun a new program that focuses on Asia's most endangered primate, the Tonkin snub-nosed monkey. We're also starting a new research program in Kenya on African elephants, which are a high priority species for the zoo's animal collection. And at the request of several government agencies, we've recently begun a head starting program for more than 80 tadpoles of California's rapidly disappearing mountain yellow-legged frog. We've also recently been approached by Fish and Wildlife Service to assist in the rescue of several endangered butterfly species, including captive breeding and propagation of their host plants. Asian turtles are the really highly threatened throughout their range, and we've begun a new research program on the beautiful but critically endangered red-crowned roof turtle in India. And you just have to love this turtle because he has an American flag on his head. And finally, we're hoping to repopulate the almost extinct population of Pacific pocket mice on Dana Point through a science-based translocation program over the next 5 years. So at the Beckman Center, we understand that conservation really is not about preserving the past. It's about working to create a future where people and wildlife can thrive together. And we depend on the support of individuals, foundations, and corporations to carry out our work, so that with the help of our conservation partners, we can continue to practice the science of saving species. And I'd like to leave you with just a very short video clip that I think sums up why this conservation work and public support for it are so important.
It is perhaps the most important question we have ever asked, and possibly the most difficult to answer. Trees, as we know, produce oxygen. Birds and insects pollinate the plants that feed us. We know that some animals need plants to survive, and some animals need animals. But why are we here? Giant whales couldn't survive without tiny plankton. Giant redwoods couldn't survive without insects. But could life on this planet survive without us? What is our purpose? Why are we here? Maybe it's because of all the species that share this world, we're the only one with the power to protect the entire planet. The only one with the power to protect every species on Earth, including ourselves. Every species is here for a purpose, and each of us has a part to play.
So thank you very much for coming tonight.